Life is something absolutely bewildering. Take any atom in your body, and it's no more alive than a grain of sand. But when gathered within the nurturing refuge of a cell, these atoms, arranged into molecules, take part in an astonishing choreography, the sum of all these dances producing what we call the miracle of life. This remains a major unanswered question. How do we go from the inanimate to the living? At what point does matter organize itself to function autonomously? To try to unlock the secrets of the origin of life, we have to start by looking at these basic ingredients which, surprisingly, are incredibly commonplace. If we look at the fundamental building blocks of life, we see that almost all the atoms that make them up belong to the Schnapps group. Because carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur together make up 99% of the atoms found in living organisms. If you add a touch of calcium, iron, potassium, magnesium, and zinc to these Schnapps, you have almost all the chemical elements that make up a living being, whether it's a goldfish, a lettuce, or a human being. But where it gets dizzyingly complicated is that these atoms are only the first building blocks of a much larger structure. Because within us, these atoms arrange and bond together to form molecules of incredible diversity and complexity. So, even if the basic ingredients are well known, the recipe itself remains a mystery. When people began to wonder how matter managed to become more complex, eventually crossing the threshold into life, they tried to recreate the first steps of this story in the laboratory. In 1952, the chemist Stanley Miller conducted an incredible experiment, the goal of which was to recreate, on a small scale, the conditions of the primitive Earth. His setup is relatively simple. Two glass flasks are connected to each other by tubes. In one of these flasks, liquid water is continuously heated. The water vapor that escapes from it travels through a tube until it reaches the second flask, in which there is a mixture of methane, ammonia, and hydrogen. This mixture was supposed to represent the composition of the primitive atmosphere of our planet, as people understood it in Miller's time. There, two electrodes subject the mixture to repeated electrical discharges, simulating the lightning storms that must have raged on Earth shortly after its formation. Then, the mixture is cooled before the cycle starts again. Just a few hours after the experiment begins, Miller notices that the initially clear water starts to turn slightly yellow, then brownish, and after a few days, it takes on a dark color. By analyzing the compounds present in this tea-colored water, Miller discovers a profusion of amazing new molecules that have formed from those that were broken apart by the force of the sparks. In this organic broth, he identifies 5 amino acids, 3 of which are used to make proteins in the cells of living organisms. It's a thunderbolt. Miller has just demonstrated that relatively simple molecules, when exposed to energy and temperature gradients, eventually form the basic building blocks of life. The excitement around this amazing experiment, which was reproduced on a small scale under bell jars, was total. For the first time, the boundary between chemistry and biology seems possible to bridge experimentally. Reading the newspaper articles from that time, you almost get the impression that now all it takes is for someone to shake it all up for life to magically emerge from this prebiotic soup. Except that it's obviously not that simple. And even though half a century has passed since then, and other similar experiments have managed to produce, under conditions like those on early Earth, almost all the amino acids used by living beings, we are no closer to synthesizing life today than we were in 1953. All these amazing experiments show that, without the intervention of any enzyme, and in the absence of any cell, simply from simple gases, water, and energy, matter can naturally form some of the basic building blocks used by living beings. But none has created life, nor demonstrated how it appeared. In fact, the gap in complexity between the molecules produced in these experiments and those that operate in our cells is such that, in all likelihood, we shouldn't even exist. It's tempting to imagine that once you have amino acids, that's all there is to it. Except that amino acids are just the first level of a gigantic structure. The real cornerstones of how cells work, which make up all living organisms on Earth, from head to roots, are proteins and the molecules of RNA and DNA. To build a single protein, you need to assemble several hundred to several thousand amino acids in a particular order. And there are several billion proteins in just one of our cells, and each of these proteins is an astonishing jewel of complexity. Consider that to obtain a collagen protein, which is found in connective tissues, skin, bones, or even ligaments, you need to arrange 1055 amino acids in the correct order. Imagine that such an arrangement could spontaneously occur by chance. It seems just as unlikely as seeing a tornado pass over a pile of metal parts and leave behind a fully assembled helicopter working perfectly in mid-flight. To give you an idea, given that living organisms use 20 different amino acids to produce exactly one medium-sized protein of 300 amino acids, there are 20 to the power of 300 possible combinations. That's a number greater than the number of atoms in the observable universe. And the vital processes that allow cells to sustain themselves and the organisms they belong to stay alive require tens of thousands of different types of proteins. Even crazier, for a protein to be functional, it not only has to be made up of amino acids arranged in the right order, but it also has to undergo a kind of chemical origami and fold in a very specific way. And above all, where it seems to get completely crazy, is that to make these proteins, current living organisms use ribosomes. These are essentially huge molecular machines made partly of RNA and partly of proteins. Logically, it's a bit like having a printer factory that needs to use the machines it builds in order to be built. For decades, this paradox of living beings operating with molecular machines that can only be made by processes involving them seemed to be an insurmountable wall in uncovering the mysteries of the origin of life. And it was extremely difficult to explain how such a complex system could have developed step by step when, at first glance, all these parts seemed necessary for it to function. So for a long time, it was tempting to imagine the emergence of life as a miraculous phenomenon. But discoveries made over the past 30 years have begun to show us that all this complexity seems less like the result of highly improbable events than the marvelous fruits of a long and thrilling history of matter, which, subjected to simple natural phenomena, can by itself take on astonishing forms. Fasten your seatbelt, relax your mind, and get ready to embark. A journey that will take you from the outskirts of the stars to the far reaches of space via the surface of an unrecognizable Earth, where we will dive into primordial oceans bathed in energy, volcanism, and raw chemistry. Back in a time when life did not yet exist, but everything already seemed to be falling into place so that it could one day emerge. Not as a miracle that fell from the sky, but as a slow and almost inevitable consequence. Our journey begins with the keystone of the Fantastic Six, carbon, the undisputed king of complexity, the central element around which all known chemistry of life revolves. The key to C's success lies in the fact that this atom has a kind of superpower, the ability to form up to 4 stable chemical bonds with many other elements, but most importantly, with itself, and to arrange these bonds in space to build molecular cathedrals, endless chains, interlocking or branched cycles, which evoke both trees and labyrinths, while other atoms quickly lock themselves into repetitive or rigid structures. The bonds formed by carbon atoms are flexible enough to allow transformations, but also stable and strong enough not to break apart at the slightest disturbance. This slight weakness of the bonds gives carbon its greatest strength, allowing it to form, through the random play of collisions, a truly staggering number of molecular shapes. Consider that, to date, more than 10 million different compounds are known in nature, and chemists have already identified, synthesized, or catalogued more than 200 million carbon-based molecules. In fact, what is called non-carbon chemistry, the chemistry of oxides, salts, minerals, in short, everything else that is not carbon-based. At best, it totals only a few 100,000 to a few million distinct compounds combined. But the real mind-boggling part begins when we stop counting the carbon-based molecules and start looking at those whose existence is theoretically permitted by the laws of nature. There, organic chemistry, which focuses only on carbon, shifts to a whole new dimension, because the number of theoretically possible molecular structures explodes to reach values beyond comprehension, on the order of 10 to the power of 100. In short, in terms of possible complexities, carbon has undeniably dominated the game. Even though its presence doesn't guarantee the appearance of life, we think that if life emerges elsewhere in the universe, it might not be universal in its forms. But it's probably still based on carbon. The incredible abilities of these atoms to create complexity have been known for over a century now. But it's only relatively recently that we've discovered they didn't wait until they were on Earth to start expressing themselves. Because their creativity shows up in spectacular environments, much earlier than we would have thought. In the swirls of gas and dust that spread and cool around stars that have reached the end of their lives, we find large amounts of carbon, which, after being created by the nuclear forge, drifts into the cold of space, where it combines with other elements and with itself to form molecules that did not exist before. The interstellar medium is not the lifeless void we sometimes imagine. Within the gigantic molecular clouds that drift like ghostly mists in our galaxy, and whose temperature is only a few degrees above absolute zero, organic chemistry is quietly at work. On the surface of microscopic dust grains that form in these remnants of ancient dead stars, the molecules of water, methanol, ammonia, or carbon monoxide that cluster together can be impacted by powerful blows. Cosmic and ultraviolet radiation that bathes space and gives rise to long carbon-based molecules. And now we see more complex chemical compounds appearing in the cosmos, such as alcohols, aldehydes, and carboxylic acids. We have even been able to identify in these clouds the presence of hundreds of different organic molecules, including aromatic hydrocarbons, or even ethyl formate, a unique compound that contributes to the flavor of raspberries and the sweet, pleasant aroma of rum, and which is regularly used in perfumery. In short, in space, as soon as you have carbon, a bit of energy and time, complex organic molecules will form. In total, in the nearby interstellar medium, and even in the more distant regions of deep space, by analyzing the light that reaches us, we've been able to identify the presence of nearly 300 different molecular species. None of them is an amino acid, and we're still very far from the chemistry of life. But it's already the start of a vocabulary. When these molecular clouds collapse in on themselves, giving birth to planetary systems, the diversity of the matter they contain will increase even more. This matter, after being transformed by millions of years of silent chemistry, then finds itself swirling within protoplanetary disks. In these concentrated and turbulent environments, a new chemistry comes to life on the surface of dust grains, stimulated by the radiation from the newborn star and by constant collisions. But the real leap in complexity occurs at the very heart of the first rocky bodies. In asteroids, the heat released by natural radioactivity melts the internal ice. The liquid water circulating inside alters the minerals and transforms simple molecules into complex organic building blocks, into amino acids and nitrogenous bases. Today, the study of comets that formed in the coldest and most distant regions of the solar system and have preserved almost intact the contents of the disk, primordial, it allows us to access the archives of that era, whose signature they have captured and preserved, trapped in their ice. By studying in the laboratory these bodies that formed at the same time as our solar system, we have discovered amazing things. Some meteorites that belong to the family of carbonaceous chondrites contain up to 5% by mass of organic matter. Within them, a large number of simple carbon-based organic molecules have been found, such as sugars, and an astonishing number of complex molecules, such as carboxylic acids, alcohols, and amines, as well as more than 90 different species of amino acids, 12 of which are involved in the formation of proteins. In samples taken from the asteroids Bennu and Ryugu and brought back to Earth, scientists were able to identify in their dust the presence of our complete genetic alphabet, the 5 nucleobases found in DNA and RNA molecules. Perhaps even more impressive, it is believed that comets reaching us from the most distant regions of the solar system contain, if we set aside the ice, up to 50%. By mass, organic matter formed by the combination of simple carbon compounds exposed to ultraviolet rays. Definitely, the basic building blocks of life were abundant in the early solar system, where many of these rocks rich in new chemical elements went to deliver their gifts to the forming planets by crashing into them. Today, it is estimated that every year about 10 tons of meteorites and tens of thousands of tons of micrometeorites fall to Earth, which is relatively little compared to the mass of our planet. But during the Hadean period, more than 4 billion years ago, this flow of extraterrestrial material impacts was probably hundreds to thousands of times higher than it is today. One might think that all this complexity crashing down to Earth from space was then destroyed by the violence of the impacts. Except that laboratory experiments show that a significant portion of organic molecules survive surprisingly well during large-scale collisions. Even better, under certain conditions, these impacts create temporary environments where the extreme temperature and pressure can give rise to new molecular species. Thus, it is now believed that the intense bombardments that mark rocky planets at the beginning of their existence are less hostile events to the emergence of life than necessary steps to enrich planetary surfaces with molecular diversity. Life did not fall from the sky, but certainly some of its ingredients did. Once deposited on the ground, these molecules from space meet those produced on the surface, and together they will cross new thresholds of organization. Because early Earth offers an extremely varied playground to test new chemical formulas. Under its gaseous atmosphere, filled with lightning and charged with vapor, water, and suspended volcanic particles. Indeed, we find a mosaic of different ephemeral environments, which appear and disappear with the tides, eruptions, impacts, and fractures of the crust. And each of these environments is a new playground, with its own constraints and opportunities where matter can display all its diversity. Each volcanic area, rich in reactive minerals, each surface exposed to intense ultraviolet radiation, are like bubbling laboratories, cauldrons of fleeting experimentation within which the range of chemical possibilities takes on a whole new dimension. Most reactions fail to progress toward life, but others succeed. And the planet acts as an evolutionary filter, selecting the molecules that will contribute to the emergence of life. But the atmosphere isn't the only factor in this change. Beneath the surface, in the depths of the nascent oceans, the molecules find crucibles for experimentation, perhaps even more fertile, nestled in the crevices of hydrothermal vents, through which some of the excess internal heat of the planet escapes. Matter crosses new thresholds of complexity. Many scientists believe that these deep refuges were stable enough laboratories for the first truly functional building blocks of life to emerge. Within them, matter, relatively protected from the turmoil of the surface, can explore complex reaction pathways and test much more ambitious molecular architectures. Because these submerged mineral cathedrals offer conditions that can give a boost to diversity. There are crevices there in which water, after seeping into the Earth's crust, comes back out at several hundred degrees, loaded with minerals and dissolved gases. And the porous mineral walls it passes through act like micro-laboratories with unique conditions. In some places, the temperatures inside these tiny compartments drop sharply over just a few centimeters. In others, the acidity varies greatly. And sometimes, the differences in charge between two nearby rock capsules even form kinds of natural batteries. To sum up, in these miniature cavities, which act like mineral protocells before their time, the confined matter is almost forced to interact with itself. The minerals circulating inside, or those that make up the walls, guide the assemblies and stabilize complex chemical intermediates. And the diversity of molecules stops depending entirely on random collisions, and instead becomes channeled by the geometry of the rocks. In short, these tiny volumes are cauldrons of innovation. And all these miniature experiments are carried out in parallel across the hundreds of thousands of hydrothermal vents which operate simultaneously on a planetary scale. Experiments that try to simulate these mini-environments in our laboratories show us that, in these crucibles that isolate, concentrate, and organize, simple molecules can generate dozens of complex organic compounds, and that around them, a swarm of other fleeting molecules can emerge. Sometimes certain conditions favor their assembly. And we see, through the processes of molecular selection, the appearance of structures that protect each other. Others spontaneously assemble into rudimentary membranes, thus creating temporary compartments where the internal chemistry differs from that of the outside. So of course, as complex as the molecules that can form here may be, this is still not life. Many of these chemical systems created in the laboratory collapse at the slightest change in conditions, but each unstable protocell, each self-sustaining chemical cycle obtained, shows us that matter, left to itself and supplied with energy, can, once again, be capable of astonishing inventiveness and brings us, even if just a little, closer to the great mystery of the origin of life. And above all, we must keep in mind that all our experiments are conducted on a small scale and over extremely short periods of time compared to the immensity of geological timescales. Yet, each of these vents could remain active for hundreds of thousands of years. This gives credence to the idea that the emergence of life is not the result of an improbable roll of the dice, but a statistically inevitable consequence of a colossal number of attempts taking place on the scale of an entire planet. Nevertheless, it must be emphasized that what we are able to recreate in the laboratory today resembles life about as much as a spark resembles a fire. And yet, experimental organic chemistry, which has reached an absolutely insane level of complexity, shows us that what matter can do on its own in its environments is truly mind-blowing. There's almost no mystery left today in synthesizing the building blocks. In the lab, we know how to produce just about all the basic molecules used by living things without involving anything living. Amino acids, nitrogenous bases, sugars and ribosome precursors, lipids and even nucleotides. All of this can emerge without any cell, enzyme or strand of DNA being involved, as long as you bring together carbon and energy under certain conditions. And what's even more interesting is that these molecules appear in a wide variety of simulated environments. This suggests that the first steps of prebiotic chemistry do not depend on a single scenario, but are the result of relatively flexible processes. Except that having all the necessary parts to build an insanely complex machine doesn't tell you in what order they should be assembled, nor how to give it dynamics of its own. Today, the big question is no longer so much about how the building blocks of life are made, but about understanding how to get this abundance of blocks to actually build something. In our experiments, we sometimes see surprising things appear, such as unstable molecules capable of catalyzing their own formation, that is, copying parts of themselves, and simple lipids that spontaneously self-assemble to form rudimentary membranes, fragile and short-lived, but capable of enclosing internal chemistry different from the chemistry outside. These membranes can even grow, absorb new lipids, deform, and divide under stress. We're still far from the cell, but chemistry has made bubbles. Once again, this is still not life, but it vaguely resembles its shadow. We know how to produce building blocks. We know how to produce unstable compartments. We know how to produce rough mechanisms of self-combustion and amplification. But we have absolutely no idea how to bring all this together in a lasting way, in a system capable of persisting, and above all, of transmitting its organization over time. It's important to understand that the main problem with all these complex molecular systems, which can form naturally, is that they are unable to reproduce and accumulate innovations over long periods of time. Their chemistry can be inventive, but it has no memory. Every complex system that appears in our experiments eventually dies out as soon as you change its environment. Yet all life can pass on its organization over time. The discovery of these sophisticated chemical reactions, which mimic certain traits of life, leads us to ask a strange question. At what point does matter stop being simply complicated and become alive? How far can matter go without crossing the threshold into life? And where is the boundary between the inanimate and that which comes to life? Being able to answer these questions would certainly earn you a Nobel Prize. For centuries, life was thought of as an essence, a mysterious property, a vital breath that animates matter. And until very recently, its origin was associated with an improbable and somewhat magical biological moment, capable of making fully formed, trembling cells emerge from steaming primitive oceans. But recent discoveries have gradually replaced this vision of a single starting moment, from which life would have emerged by enchantment, with that of a slow transition, so spread out over time that it is impossible to date precisely. Today, we actually believe that there are no clear boundaries between the living and the non-living, no precise moments when the ingredients suddenly start to work together, but rather an accumulation of increasingly complex processes which appear well before the first clearly defined cells, without ever suddenly crossing a sharply drawn dividing line. Trying to find the date when life appeared will surely remain forever out of reach, because it doesn't emerge as a spectacular event. Since it's impossible to identify a single founding moment when matter first came to life, researchers today are trying to understand how and why it managed to become more complex. The idea is less about whether the conditions were favorable or hostile to life, and more about asking whether these conditions were suitable for the production and preservation of the organic compounds necessary to trigger a transition from non-living to living. This perspective might seem to contradict the sense of wonder inspired by the complexity of proteins, which I mentioned in the introduction. But their existence is only incredibly unlikely if we imagine them appearing on the scene already fully formed and functional. However, today we believe that none of these proteins assembled all at once. Their appearance was extremely gradual and happened through imperfect molecules, shorter and unstable, and over a long period of time, evolution would have gradually transformed these rough drafts into reliable machines. So life would not have started like a turnkey factory, but more like a rickety printer, producing copies that were often flawed, but still stable enough to function and improve. And among these drafts, some that were more efficient, more reliable, and more precise eventually gave rise to the life we know today. Even though we no longer think of the appearance of life as a single initial spark, the order in which the different layers were built is still being debated. There are two major competing hypotheses that have been debated for decades to try to explain how this accumulation of inanimate chemical compounds eventually formed a little sack of chemicals that started to hiccup. The first hypothesis, the RNA first hypothesis, is based on a simple idea. Life begins when something can copy itself over time, no matter if it's fragile, slow, or imperfect. In this scenario, the appearance of RNA molecules plays the main role, because they can both encode a message and put it into action. In other words, they can store information, like DNA, and act chemically, like enzymes. If we believe this scenario, the first living systems would have appeared before the formation of cells, through fragments of RNA copying each other. Then, later on, an infrastructure would have been added around them to protect, nourish, and stabilize these messengers. The main weakness of this hypothesis is that RNA molecules are extremely fragile and unstable. To make them and be able to maintain them without being protected in the cocoon of a cell requires such specific conditions that it's hard to explain how such a sophisticated molecule could have existed without a scaffold to protect it. The second hypothesis is the metabolism first hypothesis, which completely reverses the logic by assuming that life does not begin with a code, but with a chemical system capable of lasting. In this view, for life to appear, it must first have a framework of chemical reactions organized autonomously, without the support of genetic information. And these bubble-like systems would have gradually become more complex, eventually developing the genetic code, the blueprint that allows them to reproduce. In other words, life would have started as a factory without a blueprint, before inventing the instructions to pass on what was already working. The problem here is that a metabolism without a blueprint to copy itself works, but disappears without offspring. Today, even though the question still hasn't been settled, we think that the key to the mystery of the sequence of stages lies somewhere at the crossroads between these two hypotheses, and we're trying to understand how these two logics eventually came together. If life turns out to be a process that emerges as soon as you have the right basic ingredients, compartments to concentrate them, and enough energy and time, then life stops being a fluke, an isolated anomaly, and Earth appears in a new light, that of a planet that checked enough boxes at a certain point in its history for life to appear. On a cosmic scale, the conditions that existed on the early Earth are probably not unique. Billions of planets have formed around stars similar to ours in our galaxy alone, and most of them probably inherited the same building blocks from space. From there, it becomes hard to imagine that matter remains silent on all the other planets. Today, the question may not be whether life can exist elsewhere in the universe, but to understand whether the steps that occurred on Earth are all necessary for its emergence, or if life can emerge through complexity that might have developed along different paths.
Karim, hello. Thank you for your participation in writing this episode and for reviewing my work and providing me with lots of ideas. I still have plenty of questions. I'm only going to ask you three, which I think are already quite mind-blowing. These are points we didn't really cover in this video. We often hear, and in fact we say it in the video, that life must depend on water or need water to arise. Where does the idea come from that this is considered an almost universally necessary condition today? And the second question, which is a bit intertwined with this one, we haven't really dug into this question. Why do we consider carbon to really be the prime candidate, the king of atoms for building life? And why do we consider that among the other 118 atoms, I think that's how many there are in the periodic table, of elements, we don't seriously consider others as candidates for making life in the broadest sense of the term?
Yes. Well, these are indeed very, very important questions. To begin with, water is actually something very ordinary. It's one of the most abundant molecules in the universe, H2O, made up of the most abundant elements in the universe. Hydrogen is really number one, the king of elements, abundant everywhere. Then there's helium, and after that there's oxygen, which is third in the ranking. Helium doesn't form bonds. So oxygen and hydrogen are the ones that combine the most easily, just in terms of abundance. And so water, it's everywhere in the universe. The specific thing that seems important to us for the emergence of life is having water in a liquid state. So we find ice everywhere, we find vapor, H2O gas everywhere. But liquid water for now, we know there is some on Earth. We know there was some on Mars in the past, and we're almost certain there's some beneath the surface of the satellites of the giant planets. And then, well, since it's everywhere in the universe, there's probably some on exoplanets, and I would say there are necessarily exoplanets with rivers, streams, seas, and oceans. So why do we think it's important for life? Well, first, we have an example that works with water, which is terrestrial life. And in science, it's still important not to just speculate or dream. So we have a basis, and we can study the crucial role of water in the emergence of life on Earth and the functioning of terrestrial life. And this crucial role, in fact, is explained by the fact that it's a very small molecule, 3 atoms, small atoms, HOH. It has a slightly V-shaped form. And oxygen tends to attract electrons to itself. And so it will shift the electron cloud towards itself. And so it will create a small electric dipole, a slight orientation of the molecule. And this V shape, this small electric dipole, which we call a dipole moment, will generate an ability to solvate many different molecules. There are many molecules that will go into solution. It will also play a role in reactivity. Actually, it's not just a solvent in a chemical reaction. It's not just a passive medium where things happen. It's a real participant. And actually, when we do organic chemistry, when we study chemistry, well, generally, when we do syntheses, we never use water. We work with solvents, with skull and crossbones labels, very dangerous stuff, hexane, benzene, acetone. And that's because each solvent has the ability to direct the reaction of molecules in a certain direction. And when we want to do a synthesis, we'll get a good yield. So I don't know, if you want to make aspirin, you don't want to end up with 0.1% aspirin and the rest being all sorts of other things. So we're going to select the solvents for each step of the reaction. And for life, for the appearance of life, we think that, well, there must have necessarily been a great diversity of reactions. And there was no one to say, at step 1, use acetone, then I evaporate the substrate, I put it in pyridine, and then I do 2 steps. After that, I switch to ethanol, etc. So water, being a universal solvent, allows for a great diversity of reactions. And it seems necessary to us for life to appear. Now, I'm not saying that it's absolutely impossible for life to form in other solvents, but water is going to be one of the most abundant solvents that we find in the universe. Now, we know there are methane lakes on Titan, but those are really very special conditions. Water still has a range in which it can remain liquid, and that range is relatively wide. So, as I said, it's very abundant. So that's still what I would bet on. Not ruling out that it could be possible otherwise, but I wouldn't really know what to look for or how to look for it. So carbon, well, the reasoning is kind of the same. That is, I said water is something that's common and abundant. Liquid is special, but water as a molecule, and carbon too, it's one of the most abundant elements in the universe. We observe in the atmospheres of other planets, or even on the moon Titan in the interstellar medium. If we point telescopes, we'll see hundreds of different molecules formed from carbon and very few from other elements. And that really illustrates carbon's ability to form bonds with itself, but also with other atoms like hydrogen, oxygen, nitrogen, and so on. All the basic elements of life on Earth. But this organic chemistry happens naturally, in a very diverse way, everywhere, all throughout the universe. And one of the reasons for that is that carbon is kind of the Goldilocks element. Sometimes we talk about the habitable zone, the Goldilocks zone, not too far, not too close. Carbon is the Goldilocks element because it forms bonds that are neither too weak nor too strong. Some elements form extremely strong bonds. And once the bond and the molecule are formed, nothing else happens. And others will form bonds, but they're so weak that nothing stable can be created. Carbon has this somewhat intermediate property. We know and can measure that this isn't true for the other elements. So I would bet on carbon as well, both for reasons of abundance and for reasons of reactivity. But of course, I'm not going to say right now, today, that I'm sure there is no life based on other elements. And there are researchers who are considering, for example, life based on silicon. But silicon, well, first of all, there is 10 times less of it than oxygen in the universe. The chemical variety we observe is much lower. Silicon-hydrogen bonds are extremely fragile. Silicon-based molecules are more like Si. When you say fragile, it's actually solid. No, silicon hydrogen is very unstable. Silicon hydrogen, sorry, I meant more oxygen. No. On the other hand, silicon oxygen, well, there you go, SiO2, well, that's sand, that's glass. It's the exact opposite. We're in a different situation. And then, well, same thing, but there are people who are thinking about that. But what are we looking for? How, where, we don't really know, so we can keep that in mind. But from a scientific research point of view, it's not that we're narrow-minded. That is to say, when we do science, we have a hypothesis and we have experiments that allow us to validate these hypotheses or to contradict them. And I would say, actually, if we were as carbon-based life, all the signs point to looking for carbon-based life elsewhere. If I dive into the periodic table and look for lead, which is below carbon a few rows down, well, if we were an extraterrestrial life form based on lead, then we would exist. We would be here asking ourselves questions about other forms of life based on other elements. Well, I think lead-based extraterrestrials, they would say so. We were lucky. If we're looking for life elsewhere, I think it wouldn't be ideal to look for carbon-based life. It would probably happen more like that. That's how it would go. That's it.
There's also a point we barely mentioned in the video. It's the one about chirality, which still seems like a mystery to me. I don't know if it's been partly solved. Can you remind us what this peculiarity is and how we understand it today? Yes.
So chirality is indeed something that fuels a lot of thought and research. Actually, organic matter can occur naturally, as I was saying, relatively easily in many environments. But what we'll have is a kind of soup of many different molecules. You just have to look at what we find in meteorites, in comets. Some meteorites have been analyzed, and we see millions, really millions of different chemical formulas. And there's no life. That's not life. It's just the spontaneous chemistry of carbon in different astrophysical environments. Living beings are unique because they sort and organize. And when we look at how life works on Earth, it's basically based on proteins. And basically, they're the engine behind all the reactions, the thing that allows reactions to be properly directed in how living things work. And then DNA and RNA, which kind of store the blueprint for how things work, the information. And well, everyone has in mind the double helix of DNA, which has a geometry. So not only does it store information, it also has a particular geometry. And proteins also have a very specific geometry. And all of this is built on the basic building blocks of proteins. I'll stick with the example of proteins to keep it simple. Proteins are made from molecules called amino acids. And if, for example, we create a discharge in an atmosphere that resembles some models of the early Earth's atmosphere, if we look at what we find in meteorites, we will find amino acids. But on the other hand, we will find amino acids in two geometric forms, the simplest analogy being our two hands, the left-handed form and the right-handed form. It's the same chemical formula, a palm, 5 fingers, but if I take my two hands, I can't superimpose them like this. I have my left hand and I have my right hand. If I really want to superimpose them, I'd have to tear off my fingers, so I'm not going to do that. However, they can be superimposed by taking the mirror image of one of them. And that's what we call chirality.
These molecules are made up of exactly the same atoms in the same order. They just don't have the same geometric structure. They have the same properties.
They share the same properties except in their interaction with light. And that's actually one way to detect them. But chemically, they have the same properties. And actually, let's imagine that for our body to function, we need to form proteins, but to look for amino acids, right now the genetic code says go get alanine, an amino acid. But if I randomly take alanine in the left-handed form or the right-handed form, well, I'm going to structure my protein geometrically differently. And so it's a disaster. That is, by saying go get alanine, then this other amino acid, then another amino acid, I have a 1 in 2 chance of getting the right one. And in the end, I have 0 chance of getting the right protein because what determines the structure of the protein and its reactivity is really its shape. So in terms of natural selection, at some point where life, we don't know exactly when, but life found that it's much more practical to have only left-handed alanine rather than having a 1 in 2 chance of making a mistake or needing an extra instruction. Go get alanine, specifically left-handed alanine, or next time go get right-handed alanine. So it's a simplification that makes all life possible. And it's the same for RNA, the double helix. Well, it's based on sugars, deoxyribose for DNA, which is right-handed. Well, your helix wouldn't twist in the same direction if we only had the left-handed form, and it wouldn't twist at all if there was a 50-50 chance of having right-handed and left-handed forms. It's as if you had a kind of workbench with screws and bolts. And with 50 of your screws turning one way, 50 the other way, and the bolts the same, and then you take two of them, and actually every other time you don't have the right thing because they turn the same way, and that doesn't work. So where does it come from? So did we start out 50-50 at the beginning, and is it just pure chance? There is an explanation that fascinates me. It's not necessarily the right reason, but in any case, it follows a scientific thread that's quite dizzying. In certain meteorites, it has been shown that the amino acids found there have, for some of them, an excess of the left-handed form compared to the right-handed form. It's not 100% of the left-handed form, but there is 10 to 15% more than the right-handed form. And so if on Earth it was this type of meteorite that brought amino acids, well, just the fact that there is a little bit more of one form, that there was a little more of one form than the other, made it more likely for life to eventually select through evolution this configuration. And this success, and that's what I was saying was mind-blowing, so that's an observation, we have a small excess. And we explain it by irradiations that could have happened in the molecular cloud before our solar system formed. And there are certain neutron stars that spin very fast around their own axis and at one pole towards the North Pole. They will emit UV radiation, which is actually light radiation in general, which will be polarized in one direction and will preferentially destroy one type of amino acid, and in the other direction it will preferentially destroy the other type. And then at the equator, there's a mix of both, so it doesn't really do much. I'm adding a lot of ifs. But what if for life to appear on a planet, there needs to be an initial excess? And what if this excess? Would it have to be inherited from chemical processes that took place even before the star and the planets formed? And that this chemistry is linked to a neutron star happening to pass by in a particular orientation? There we really have a dizzying chain of connections.
That's not good for the emergence of life.
So, same thing, the universe is vast, but it still remains within the...
It remains within the realm of possibilities.
Yes, but it's a line of inquiry that's really grounded in science, because all of this is based on observables, on observations, on simulations that are done in the laboratory. And then maybe we have a racemic mixture. It works every time, but I can't say. Does it work every time? Is it very rare? But that's what makes this field of research, and I find it really fascinating.
Well, you convey it very well. It's mind-blowing, it's crazy, it makes you want to know more. Thank you for this conversation, Hervé, and for all these questions and all the avenues you've opened up. See you very soon. Goodbye.